So next speaker is Dr. Reyk Horland. He is the CEO of TCU's, a German biotech company which has developed a multi-organ chip technology aimed towards conducting preclinical, systemic safety and efficacy testing of substances using a combination of different human tissues. Over the course of his academic career at the German Arthritis Research Center and the TU Berlin in the group of Professor Roland Lauster, he has pursued the development of tissue models that can mimic human biology in vitro. Since joining TCU's in 2010, he has led the efforts to establish and characterize a chip-based vascular model in an interdisciplinary team of engineers, computational modelers, and biologists. His talk today will focus on applications of new multi-organ chip tools for safety and efficacy assessment.
A very good morning or good afternoon, I guess it's in India, to everybody. My name is Reyk Horland, and over the next 30 minutes or so, I would like to give you a little bit of an introduction on tissues technology and how the technology is employed in drug development, but also in other areas and other industry segments like cosmetics industry. Now, when it first of all, a little bit of an introduction to the company. I guess as many of the other presenters from industry, the Tissues is a spin-off from the university, from a technical university in Berlin. We were founded already in 2010, mainly back then as a kind of vehicle for legal matters. We are actually only active on the market since around 2014 and 2015. We are currently around 32, 35 employees and still have a very strong and good relationship with our mother institute, which is the Medical Institute of Medical Biotechnology at the university, where an additional 30 associated researchers are working for us. Since then, we were actually able to build quite a nice customer base, both in industry and academia, and we are very proud that we are certified under ISO 9001, which means that we have a total quality management system in place, which we believe is very important, especially if you want to bring these chip systems, these organ chip systems, microphysiological systems later on also to a qualified and validated stage. Now, I don't need to tell you a lot about this, so I'm just going to skip that slide. You know, that's the usual slide explaining you why we need to develop and invest in other methods than the conventional cell culture technology or the animal models. For us, I will go a little bit into more depth about that in my presentation on Friday. We even want to go one step further and go into the direction of personalized medicine and kind of potentially clinical trials on chip with a combination of iPSC-derived models and microphysiological systems approaches. Now, when it comes to our technology, our multi-organ chips, like most of all of the other MPS solutions which are available, you know, they usually consist of two main components. First of all, of course, you need to have the chips. We do have a number of different chips, and of course, you need the organ models, the biology in the chips. And as you can see, we do have a very broad portfolio of different organ models, which we have established on that chip solution. It is important here to say that we do not want to reinvent the wheel. So the chips are quite flexible, meaning we do not have specific chips if it comes to, you know, modeling the liver or to modeling the lung. Instead, the chips, they differentiate from each other by the number in the name of the chip here, which will always give you an indication on how many different organ models you can combine on one chip system. So this chip here would have like an option of combining two different organ models in one common circulatory system, this one free and this one four. So this is the flexibility of the system, which did allow us actually to model that many different organ models system, and the sources of these models can vary. So sometimes we do have models which are built based on cell lines. And why do we still use cell lines? Because in certain contexts of uses for a certain fit for purpose, these cell lines are sufficient. And I will also later on give you an example of when we use the cell line, just because the functionality of the cell line for that specific context of use was sufficient, and we didn't need to use, for instance, which is another option, primary cells is a source for these models. Other sources I already mentioned that could be iPSC-derived models. We can actually work directly with biopsies or precision-cut tissue slices in our systems. And coming back to my saying that we do not want to reinvent the wheel, you can also work directly with the cell culture models which you may have developed in your laboratories, or which are available from commercial vendors in our systems. So, if there is a best-in-class model, it can basically be used in our chip system. And then, in order, to run the features of the chips, need to have certain devices which are shown here. So this is just, again, an overview of the different chips we have available. You see, depending on the chip type, the microfluidic design can be a little bit different, up to a very complex design, which you can see here on the right side of the screen. Now, with these chips in hand and basically the models which we have, you can now think about setting up assays. I already mentioned that you do have a lot of different options which can be explored if you want to build certain models on a chip. For us, then, of course, especially if you think about a multi-organ approach, there's always a little bit of model preparation time required, which is usually done outside of the chip. So we mature the models outside of the chip, and then we bring them together in the chip. What we see then is that, if you bring a cell or a tissue from a single culture into this kind of multi-organ system, there is some time around 24 to 48 hours where it needs some adaptation, because all of a sudden, there are so many different cells in the tissue or in the chip, which all can interact with each other because they are interconnected by this microfluidic circulation. But then the whole chip enters a homeostasis, and then that's when basically you can start doing safety and efficacy. We usually aim for, let's say, run times which are designed to also include repeated long-term exposure of the system. Sometimes this can be just 14 days. In this case, I'm writing here 4 weeks, which is our usual time. But we also have assays, and I will show you also an example later, which runs sometimes for up to 2 months in the system. And the first goal is always to have these models established in the system, in the chips, which are then functional and viable over these 4 weeks of time. And then you can think about how do you want to, what is your scheduling, your dosing? Do you want to go for a single dose? Do you want to go for repeated dose with recovery phases? Do you want to do dose escalation? Because you have such a long time, if you have such long experimental time, you can of course play around with the exposure regimen. During the experiments, you can take samples out of the chip. That varies a little bit depending on which organ models you have in there. If you have mainly organ models which you use kind of in, let's call it standard submerged culture, you of course have a little bit more sampling volume available than if you think about barrier models, especially barrier models like the lung and the skin, which we cultivate on the air-liquid interface. in the chip, which means basically there's a little bit of less volume, total volume in the chip. But in general, in average, you do have per sample point around 250 microliters per chip available, which gives you kind of some flexibility with regards to the analysis which you want to do, like very basic parameters, glucose, lactate, LDH for basic toxicity, but you can also go for ELISAs, for cytokine measurement. You can go for metabolite analysis via LCMS, for instance. Then, of course, the chips are also fully microscopable, and the more non-invasive kind of an control, an in-process control or an endpoint control is, the better it is for the system. So you can do a lot of things with imaging, just looking at cell morphology with bright field imaging, but also, putting tracers in there, doing confocal imaging, for instance, looking at migration of cells. So there's a lot of options with regards to also sophisticated imaging methods. And at the end of the experiment, as flexible and as easy as it is to bring in different tissues into our chips, you can actually also remove them, the tissues, again out of the system, with the exception of endothelial cells. So what we can do is basically that we can cover the endothelial, the microfluidic channels with endothelial cells, and that means that you have some kind of artificial vasculature system in place. So these cells we usually, analyze, or if it comes to staining directly in the chip. Of course, you can, also lyse them and flush them out for PCR. But all the other tissues you can basically remove intact out of the chip and then go for lots of different endpoint controls, which I've summarized on the right side of the slide, like histopathology, qPCR, metabolite analysis, or any kind of omics. Now, this is kind of our basic template which we use to then, establish these assays and also fine-tune the assays which we have in our hand. And in the second part of my presentation, I would like to actually give you a little bit of an idea on a variety of different assays which we have established. And I will start in terms of, let's say, the number of organs low. So this is actually, even though we are always, having multi-organ chip systems, you can also use them for single organ modeling. In this case, it's a bone marrow on a chip. And then the next examples will increase the number of organs which are combined on a joint platform. Starting with the bone marrow on the chip, what do we want to do and how do we model the bone marrow on our chip system? So the bone marrow is a porous, in its core is a porous structure. So first of all, we needed to find something on how to model the porosity of the bone marrow. And in order to do that, we chose a ceramic scaffold, which is coated, it's chromium oxide scaffold, which is coated with hydroxyapatite. And this is actually a system where you can seed human mesenchymal stromal cells in, and these cells like it there and basically form their own extracellular matrix, which you can see in these kind of pictures. So you basically give them around 7 days, they attach to the surface of the ceramic scaffold and then start to form their own extracellular matrices. So kind of basically covering all the surfaces of the ceramic with their own ECM. And once that happens, you can actually then bring in the hematopoietic stem cells. And as you can see here, basically these hematopoietic stem cells then attach to the extracellular matrix which the mesenchymal stromal cells have produced, and they really like it there. So in a way, this mimics the hematopoietic stem cell niche. And if we keep the system running just with the standard cell culture conditions, we basically have a system in place where the cells in the niche are staying in terms of their character as a hematopoietic stem cell, but there's also, of course, spontaneous differentiation going on. And what happens, I'm just going to move it back here to this slide, is basically that cells, as it's shown here, can spontaneously migrate out of this bone marrow, and they differentiate when they move out. Basically, they are then in the circulation, and we have the second compartment of the CHIP2, which is left empty, where basically a lot of these cells are then sedimented, and you can use this compartment to sample the cells and then use them for analysis. But in general, the number of cells, the number of hematopoietic stem cells which are kept in the bone marrow, this stays stable over the time. However, if you change the media, you are then also able to drive the differentiation of these hematopoietic stem cells, which migrate out of the niche into different lineages, which means that you can drive the system into producing different cell types just by changing the media. And this is true for instance, for the myeloid lineage, for the lymphoid lineage, for the erythroid lineage, and you can also actually have a media which is capable of, producing mixed lineages. And most of the analysis, as you have guessed probably from these pictures already, is being done by FACS analysis. And this is just to give you a little bit of an overview of the different used in detecting different progenitors or the differentiated cells in the system. And this is just to give you an impression on how this looks like. For instance, if we start the erythroid lineage maturation on what you can see over the days with regards to differentiation of erythroid lineage in the system. Same goes for granulocyte differentiation in the system. And of course, you can also detect other populations in the systems, always over this period of time of around 24 to 31 days, and by using this certain specific media. Now you probably think, okay, what can I do with the system? And basically, this system is currently in use with a number of industries with regards to understanding bone marrow toxicity. Because basically you have a setup where you have control, so the healthy bone marrow without any substance or drug exposure, and then on the other hand, you have the exposure of the drugs, and by using these different differentiation media which we have, you can then compare the differentiation potential of the control bone marrow to the drug exposed bone marrow, and basically understand, do I see, impairment in the potential of generating lineage-specific cells in the bone marrow? And this can be, for instance, very important if you think about chemotherapy for cancer patients. So nowadays, chemotherapy is a very complex approach where you just not only give one chemotherapeutic agent, but maybe two. And the issue here is that it's not as straightforward as it sounds, that you, for instance, think, okay, each chemotherapeutic agent in itself has a specific role, and role A plus role B is A and B. This is not true. So you actually see differences, for instance, just by saying, okay, which chemotherapeutic agent do I give first? Do I start with A or do I start with B and have a recovery phase and then give the other one? And all of this, you can now use the system to understand what is the best dosing and scheduling for these kind of combinational therapies, actually before you enter the clinic and before you have to do a lot of these trials on volunteers, on patients who are enrolling into these clinical trials, for instance. So dose range finding, refining of treatments, dose scheduling is actually very, very often used, or is this assay very often used for. And of course, chemotherapeutic agents usually are small molecules, but you can also think about using the model with new approaches like cell and immuno-oncology treatments or antibody-based treatments. So that was the first example. Now the second one is actually coming from the cosmetics industry. We are increasing the number of organs to 2, and here in this case, we are combining a liver and a skin model for toxicokinetics and toxicodynamics. And this is how the approach looks like. On one hand, you have a skin model, in this case, a commercially available model from Matec, which we culture in one compartment on the air-liquid interface. So the surface of the skin is exposed to air. And in the other one, we have a liver model. And this is actually the example which I mentioned previously, that you build a model based on a cell line, because with regards to the functionality, in this case, in this context of use, the cell line is performing well. And then you can compare topical application of a substance. So you put it on top of your skin, basically, versus, let's call it an intravenous systemic application, where the substance is injected directly into this circulation on the chip. And first of all, we started very basic. We said, let's just take a compound which is an ingredient for many cosmetic product. It's retinoic acid, and we compared topical versus systemic, as I mentioned, but also single versus repeated, and then analyzed the metabolites which could be found in the circulation. And as you can see, the metabolites are quite different because the application route is different. In this case, the retinoic acid, first of all, has to pass through the skin. There may actually also be first-pass metabolization effects in the skin. So you see actually a completely different metabolite profile of retinoic acid depending on the administration route. We did the same for permethrin, as you can see again, especially if you also think about systemic application and single versus repeated, you see there are different metabolites coming up depending on whether you give it as a single dose or as a repeated dose. And a very important point, and this is also one point I will make in my Friday presentation, is actually how to qualify such a system. And in this case, actually, we did the same experiments in 2 labs. So in our labs and in the lab of our collaboration partner. And as you can see here, the data is nicely correlating, which means that we have a system in place where people in other laboratories can just take the protocols and perform the same experiment, and they will come to the same results and reach the same conclusions. So that is a certain quality standard which we always want to achieve for every of our assays which we establish. Now then we said, let's try something different. And this is another test compound, HAT, which is part of a lot of hair dyes which are on the market. And again, depending on the administration route and also on the dosing, you see that there are different metabolites coming up. We also then again wanted to take it one step further, because what we did so far was, you either give the compound IV or you give the compound on the skin and leave it there, but that's not how you apply a hair dye. You usually apply a hair dye just for a certain amount of time and then you wash it off. And that's what we tried to do here. So we again have the IV application in red. We have the topical application where we leave it on the skin. And we have the topical application where we wash it off after 30 minutes. Like you wash off, you go into the shower and wash off your hair dye. Basically, that's what we did. And again, depending on the application, or specific applications, you see different metabolite profiles coming up, which is for cosmetics industry very important in understanding safety of compound or an ingredient which is part of their product. Okay, and then Last but not least, the most complex example I wanted to show you, basically a chip-4 system combining an intestine, liver, kidney, and a brain with a blood-brain barrier. So this is data which is part of the EU Tox Risk Consortia. And in this, a couple of collaboration partners which actually do have their own tissues, coming back to my previous point, that basically you're not stuck with the tissues which we have in our hand, but you can use tissues, all other tissues too. So in this case, we combined an intestinal model from Matec with a liver model from Insphero, and a neuronal model from Biotalentum, and used these cells, the iPSC-derived kidney cells from a group of Paul Jennings at the Free University in the Netherlands for our kidney system. Now, the chip itself is a little bit more complex. You do have actually two circulation systems on the chip. Again, in red is kind of the blood surrogate circuit where you have most of the organs in there. And then you have an interface, and that interface is where our kidney model sits, basically with a proximal tubule part and the glomerulus part. And then you have this yellow excretory circuit. So basically, the kidney here is able to excrete substances from this blood surrogate stream into this excretory stream, but also to reabsorb the substances into the blood surrogate stream. And the first thing with this chip, which you have to do, is basically close this kidney barrier, because we are using different cell culture media in the yellow and the red circulation. And that's why the kidney model starts at day -7. It takes some days to completely close this biological barrier. So the kidney cells are forming a biological barrier, and only when this is closed, we basically add the other organs, the cell culture media here, and then you can start the exposure of the system, like oral uptake through the intestinal barrier, has to pass through the intestinal barrier, reaches the liver, is metabolized, and so on. The chip system itself also has an underlying PBPK model in order to be able to understand or to extrapolate the data from this in vitro situation to the in vivo situation. Just in the interest of time, I'm not go very much into detail, just telling you that basically the modeling approach is based on compartmentalization of the chip. So you basically try to compartmentalize the chip. For instance, the pump can be a compartment, the channels, the chambers, and the organs. And then there you have a definition of the compartment, and basically this model then allows you to include active transport, diffusion, even PDMS plastic binding, if that can be an issue, and protein binding in the system. And that is basically the basis to extrapolate then this in vitro data to an relevant human in vivo situation. Now, I mentioned already the first important point is to close the barrier of the kidney cells, which we usually try to understand via, for instance, FITS-inulin assays. The second one is also to have a barrier, a perfect biological enclosed barrier in place for the intestine. And for that, we tested with haloperidol, which is an antipsychotic medication. And you basically then can check if you give that intravenously. Sorry, we are still at the kidney barrier. If you give that intravenously, what kind of metabolites are coming up, and is this matching kind of with the profile which you see in the clinics from patients who are giving this medication, and that matches very nicely. And now we come to the intestinal barrier, which is the second biological barrier in our system. I already mentioned you can basically give compounds orally, meaning you apply it on the apical side of the intestinal model. It has to pass through to the intestinal barrier. There can be first pass metabolization in the intestine, and then it reaches the circulation, is transported to the liver, and is also there metabolized. And basically, with the metabolite profiles which we have in our hands, we could show basically that the intestinal barrier is intact. So really, there's also active transport of this compound through the intestine, not only passive diffusion. And the profile again, which is then the metabolization profile being done by the liver, matches very nicely what we see in the clinic. Now, this was a system basically where we used a lot of different models from different providers, meaning there could be different donors, which were the source for the different models. But can we also build a system which is derived from a single donor, which basically would a little bit go into the direction of personalized medicine? Because if that donor would be you, we would basically build you on the chip, and we would get some information on how you specifically are reacting to a certain medication. And that is what we tried here. So in this case, we tried to build all the different models, iPSC derived, from a single donor, and basically then put them together on the chip. And for the last experiment, we basically used an...
Excuse me, Herr Horland. Yeah. If you can bind up, it's good. Maybe 2-3 minutes you can bind it up. Okay.
It's my last slide. We used three different compounds in order to also understand if the third barrier, the blood-brain barrier, is actually intact. And these are atenolol, carbamazepine, and verapamil, and they are known to be like low blood-brain barrier permeable, medium, and high. And again, the profiles match very nicely what you see in the clinic, meaning we have a third kind of biological barrier which is intact. So the kidney, the intestine, and the blood-brain barrier, where you can basically now understand what is going on in such a complex system for compound. Okay, and with that, I'm at the end. I just wanted to take the last few seconds of my presentation to invite you to our second Microphysiological Systems World Summit, which will take place in Berlin next year from June 22nd to June 30. It's definitely a great opportunity. The first one, it was this year in New Orleans. It was really, really great. Lots of interaction, lots of exchange, and we are expecting even more people next year in Berlin. So hope to see you all there. And with that, I'm at the end of my presentation. Thank you very much for listening to me for I don't know how long. I'm happy to take any questions which you may have. Thank you.
Thank you very much. It was very enlightening talk. Now let us see if audience has any questions.
PD studies are relevant in real scenario, and who will pay for the cost of the testings you are conducting?
So basically, we actually do a lot of PK/PD studies, and the benchmarking is, of course, always with regards to clinical data. So what I showed you with the last example, the selection of the compounds which we used was actually being done by the industry advisory board of this EU tox risk project. So they basically selected the compounds which we should benchmark the system, because there was clinical data available where the data from the chips could be matched with and basically benchmarked with. And with regards to your second question, basically if somebody gives us a contract to test something for them, then they of course pay for that. The idea here is to say, okay, there are definitely in vitro systems, specifically microphysiological systems in place, which can give you very valuable data which are otherwise not available by using conventional cell culture method or even animal methods. And that's when also people are not afraid to take some money into their hands and purchase the services, or just purchase our technology and run their experiments by themselves. But I will go into a little bit of more details on the commercial side in my presentation on Friday also.
Thank you for the good talk, sir. I have two questions. First one is regarding the oral administration, what you spoke. So on the chip, there's only intestine, but the upper part of the alimentary canal is missing. So when you administer something orally, then it undergoes degradation or whatever it is exposed to first upper alimentary canal. So what are your comments on that? Then second question is regarding the systemic administration. So when you mean systemic administration, what is actually in the flow, whether it is a whole blood or simulated media? And if it is blood, then is there any purification system or like any other external things which is attached to mimic the in vivo condition?
Yeah, two very good questions. So the first one, basically, you can basically use a kind of, if you want to mimic what is kind of, reaching the intestine, you can pre-digest things. There are also systems available which actually do quite nicely. So you end up with a digestive juice, which is very reflective of what is entering the intestine. So that is possible if that is a valid approach for the context of use of the assay. The second question, systemic means basically you just inject it into the circulation, so in a way intravenously. It is at the moment, we're still working with cell culture media. In many cases, cell culture media which does not include FCS anymore, but sometimes, for instance, for this autologous CHIP4 approach, it's just cell culture media with a little bit of human serum. It's not blood yet, because for blood you would need to be able to have a fully closed kind of vasculature system. At the moment, we just have the endothelial cells lining the channels which interconnect the organ compartments. We do have approaches where we kind of also can form a vascular bed in these organ compartments, but we do not yet have fully vascularized tissue models which are interconnected then to these larger channels which are connecting the compartments. And that is a requirement for using blood in the system, a fully closed vasculature. Otherwise, you would just bleed blood into the tissues, and internal bleeding, is never good for any kind of mammalian system. So that's something which we need to avoid. But if that is possible, this kind of fully closed vasculature, we are actually planning to use blood or plasma in the system. We do not think that you will need a cleaning system, because I showed you the bone marrow system. Basically, the bone marrow will be able, with the cells it can produce, with the macrophages, and also with the production of new blood cells. That is the kind of, long-term idea, that you have a producer of blood cells in the system which also can clean up the old cells to keep the blood-producing cells or the blood cells in homeostasis. As long as that is not possible, you just have to have access to blood samples from that, for instance, iPSC donor, which you use to build all the different iPSC-derived models from, and use that blood, for instance. But that is kind of the long-term goal. At the moment, I believe most of the people are still using cell culture media in the system.
Excellent talk, Dr. Subramaniam from Reagent Biosciences. I'm quite impressed with your multi-organ chip, and I think you made, this was my vision back in 2000, but there are lots of barriers there, and you've made good progress. I congratulate you on that. But here, there's many other issues that probably cannot be addressed by microphysiological systems. If you go to drug discovery guys, they'll make all the compounds insoluble. They're greasy compounds. You can never make them absorb or anything, I mean, into the even systemic circulation, even rat, a dog, a monkey. And those kind of compounds, now you're putting in an aqueous solution in a in vitro system and expect them to travel into all your multi-organ systems is impossible, number one. So the drug discovery chemists have to improve on the solubility of their medicinal chemistry programs. That's not going to happen for you or me who are in this field of building these models. Number two, there are other issues, several issues. We've actually developed for industrial toxicants in 1990s, PBPK models. When I went into the pharma industry, nobody was interested because the amount of data you need to generate to build a PBPK model, you're going to be spending 2 years to develop for each compound to do it.
Sorry, excuse me.
So these are all the practical problems. How are you going to address them?
Excuse me, requesting ask a question. Let us not discuss here. It is a question session.
Right.
Ask a question.
But this is the question for him, okay? These are all the problems that are yes,
yeah, kindly, we have others are waiting actually, so.
So thank you very much for your comment and your question. Yeah, you're absolutely right. I mean, everybody who's saying that microphysiological system can basically replace kind of any animal test, I would be very careful with that. You pointed out some limitations. There are other limitations. Yeah, these systems will never be able to have emotions, feelings, or so any kind of behavioral study is not going to be possible. Any kind of study with regards to bone healing is not going to be possible in such a small system. Also, modeling a full organ or the full cardiac power of a full heart will not be possible in such a small system. So there are definitely limitations, but there are also lots of opportunities here, because if you think about I'm not talking about small molecules here, because if you look at pharma, the pipelines for small molecules are slowly, getting smaller, while new treatment approaches like antibody-based or cell and gene therapies are increasing in the pipeline. And this is something where you rarely find very good animal models about. And then also you don't run into these problems, what you mentioned, with cell and gene therapies. But you need, for these approaches, you need human systems, because these are very, very human-specific treatments, very often also involving the immune system. I believe that's where the entry hurdle for many of the upcoming also microphysiological systems will be the lowest, because the need for new models will be the highest there in the pharmaceutical industry. And with regards to cosmetics, I mean, in Europe, also in other countries, you are not allowed anymore to work on animals when it comes to new cosmetics ingredients, so they have to have alternative systems.
Thank you.
Thank you.
Hi, I have two questions. One, what kind of material is used to develop these kind of multiple organ on chips? And other question is, as you're heading towards personalized medicine, do you think incorporating patient serum to look at the immune response system is important? What are your thoughts on it?
Thank you very much for your two questions. So first of all, we do have now two types of materials which we use in the production of the chips. The first one is kind of probably known to everybody. So we have microfluidic chips which are based on PDMS. In our case, it's always kind of the lowest layer or the bottom is a glass slide, which is bonded to a microfluidic PDMS layer, and then which is again bonded to this adapter plate, which holds all the kind of fittings and connectors to the pump. And then we have another chip type, because you hear a lot that PDMS has a lot of disadvantages, which is true, especially when it comes potentially to the absorption of small molecules. But on the other hand, of course, it also has lots of kind of advantages, like rapid prototyping. You can rapid prototype new chips. It is better oxygen permeable than than plastics, for instance, which can have a huge impact on your cell culture in the chip. Especially if you think about complex cell cultures as we do as multi-organ, where a lot of different cell types need oxygen in the system. But we do have other chips which are based on COC, which is another polymer, which is basically in order to avoid or to at least reduce drastically the effects of potential molecule absorption into PDMS. And the second question is, you are absolutely right. I tried to hint a little bit on that. If you have, for personalized medicine, definitely it would be good to have access to blood or plasma of that patient, to use that also at least as a start in these kind of systems. Yeah, that would be good.
Thank you.
Yeah, thank you very much. Thank you. Let us give a big round of applause. Thank you, Professor Horland. Yeah, bye-bye.
And see you on Friday. Bye-bye.
Thank you for chairing this session, Dr. Rath.